Okay, so are you latching on to all this stuff? Here's a chance to test yourself, just like the T cells and the B cells do. You have to go through testing, all right? So, which of these statements is false? Antibodies bind to antigens and immobilize them. Helper T cells activate both B cells and cytotoxic T cells. The response of the specific immune system is exactly the same every time. B cells and T cells are made in the red bone marrow. Antigens can have multiple epitopes. So which of those is false? So the response of the specific immune system is not exactly the same every time. It differs according to what you've been invaded by. The innate immune system is exactly the same every time. The specific immune system, it depends on what you've been invaded by, hepatitis, measles, whatever, all right? Okay, so we've said that the cytotoxic T cells are killers. How do they actually kill? Well, here you see in the upper right, there's a cytotoxic T cell killing that poor little kitten. Cytotoxic T cells, ruthless. Oh my God, they show no mercy. So cytotoxic T cells directly attack infected cells. So any cell in your body that gets infected by a bad guy, cytotoxic T cells are going to kill it. Remember shooting that guy through the head in the last one, PowerPoint? Okay, so they have the lethal hit mechanism. They dock on the cell with the antigen MHC1 protein complex. They release perforins and granzymes. So perforins, once again, are little things that penetrate the cell. They perforate the cell. They punch holes in the cell. And then granzymes are what induce apoptosis. So again, cell suicide. Cytotoxic T cells convince infected cells to commit seppuku. All right? They also release interferons, which decrease viral replication and activate macrophages. All right? So the interferons then are going to be received by nearby cells all around. The T cell has found an infected cell, so basically it's warning all other nearby cells, you guys, watch out, there are bad guys here. If you're not infected, then start, preparing for a battle. If you are infected, then kill yourself. And they release tumor necrosis factor, which kills cancer cells, okay? So I said before that natural killer cells were like your body's ninja. Well, T cells are like, cytotoxic T cells are like your body's samurai. Right, lots of weapons, very lethal. Oh God, you don't want to encounter a samurai. Those are badass dudes, okay? And then here, attack of the cytotoxic T cell. Here you see they're attacking that poor dog up there. And there's a cytotoxic T cell disguised as a cat attacking that dog down there. But that illustration on the left, that's a good one. Take some time, go through it, look at that. That's got really good info. It's a chance for you to self-check and see your own understanding. There, see the perforins, granulysins. I said granzymes, that's, well, yeah, so there you go. On both sides, you see it. Here you see a T cell docking with a cancer cell, and then the T cell has caused that cell now to undergo apoptosis. Cancer cell is now killing itself, all right? And so memory cells, let's talk about those. So T memory cells, following clonal selection, some T cells become T memory cells. So remember, when the APC docked with the helper T cell, the helper T cell caused there to become other kinds of T cells. So cytotoxic T cells, suppressor T cells, and T memory cells, all right? So the T memory cells are long-lived. Unlike most of your other white blood cells, they live potentially for years. They exist in higher numbers than naive cells, so you've got bunches of these out there. And what happens then if the same pathogen tries to get into your body a second time? Upon re-exposure to the same pathogen, memory cells launch a quick attack. So look at that graph down the lower left-hand corner. You see that magenta line? Is that magenta maybe? Starting over there at day 0, first infection of antigen A. Notice how it takes a while for your immune system to see the primary response to antigen A. It takes a while. all this stuff, different, cloning all the cytotoxic T cells, cloning the B cells, the plasma cells, cranking out all the antibodies. It just takes time for that to happen. So the very first time you get infected by a pathogen, it takes a while for your immune system to completely ramp up the response and win the battle. So as you see the curve going downhill, that means you've won the battle. But then look at what happens. Secondary response to antigen A. The second time that same antigen tries to invade your body, or the 78th time, or the 364th time, or the 26,941st time, your T memory cells are going to spot it instantly, and they cause an immediate, drastic, overwhelming response so that you don't get sick again right away. Have you ever had a day like where you woke up, and one of your friends called you and says, hey, let's go shopping. And you're like, I don't know, I just don't feel very good today. Your friend says, are you sick? And you go, oh, not really. I mean, I just don't really feel exactly right, And then like a couple hours later, suddenly you're feeling fine again. You call your friend back and you go, shopping, girl, let's go. I can't say for sure what that might have been. That was a day when some infectious agent that you already defeated once, maybe in your childhood, it tried to attack you a second time. And the worst that happened was for a couple of hours, you didn't feel, fully like yourself, but then a couple hours after that, you were ready to go again. See, that's what's going to happen here. You're not really going to get sick for a week with it again. It might be, you might feel, just kind of a little for a couple hours and then you're fine, all right? That's why memory cells are such a big deal. All right, moving on to the humoral immunity then. So that was cell-mediated, that was T cells. Humoral immunity. Okay, so the recognition has to happen. So the B cell receptors bind to the antigen, take it in and digest it, then display the epitopes on its MHC2. So remember, B cells function as APCs. And remember I pointed out there was you can see here in the lower right. See how B cells have what look like antibodies on their surface? Well, essentially they are. Those are the receptors. We said that when the cells got trained, in the thymus for T cells or in the red bone marrow for B cells, they had the receptors inserted into the cell. Well, so the B cells have those receptors inserted. But when the B cells then turn into plasma cells, which you see there in the lower right, then what it does is it starts making antibodies that are exactly the same as the receptors it had on the outside of its body. So whereas the B cell by itself can grab bad guys and digest them and work like an APC, well, it is an APC, shows it on its MHC2 to the T helper cell, it also then will morph into these plasma cells that crank out antibodies so that the antibodies can wander through the body. Why is that an advantage? Well, the antibodies will exist in massively large numbers. And what they're going to do is they're going to be handcuffing bad guys. And once you handcuff the bad guy, then the bad guy can't do anything bad anymore. So it's just a way of having an incredibly effective response. The B cells will be killing them right away, but for anything that doesn't, get encountered by a B cell, well, at least there's a chance that an antibody will get it and at least handcuff it until some other kind of, a B cell or a natural killer cell or something else gets there to kill it, okay? So after activation by a T helper cell, B cells will divide repeatedly and differentiate into many more B cells as well as plasma cells, which produce antibodies specific to that antigen. So when activated by the T helper cell, B cells make more of themselves, and then they also, some of them, turn into plasma cells, which make the antibodies. Plasma cells secrete antibodies for 4 to 5 days at a rate of 2,000 per second, and the antibodies are the same as the receptors on the B cell, which we're going to learn are what are called IgDs. We're going to see that there are 5 different kinds of antibodies. So IgD is the one that's on the B cell. So they recognize first, then they react. So antibodies bind to an antigen, render it harmless, don't directly kill it, but instead tag it for destruction by cytotoxic T cells, natural killer cells, and other white blood cells. So again, antibodies are so great because there's just jillions of them floating through your body fluids, And they're encountering bad guys like crazy, and they're just handcuffing the bad guys immediately. We say that they mask the pathogenic region of the antigen. So what that means is when they handcuff it, they prevent that antigen from doing any more damage, all right? So the antigen is still not killed, but at least it can't do any more harm. It's kind of like when they handcuff them, they also take their weapon and disarm them. So the antigens can't do any more damage once they've been handcuffed by an antibody. And then they also remember, so some of the B cells will get cloned into B memory cells. So you have both T memory cells and B memory cells, long-lived cells that are going to try to make sure that if you ever get invaded by that same thing again, it's not going to even have a chance, okay? All right, so the attack, what happens in terms of attacking? So antibodies, as I said, mask the pathogenic region of the antigen. They render the pathogen helpless, and then they attract cytotoxic T cells, natural killer cells, eosinophils, whatever, okay? So you see in the upper diagram there. Yeah, tag them. There are complement proteins tagging them as well, all right? So the whole immune system now comes into play. Once that helper T cell gets alerted by an APC that bad guys are out there, remember, it basically mobilizes your entire immune system. Natural killer cells, neutrophils, eosinophils, complement proteins, antibodies, everything is now going to work. When we talked about the lines of defense, it's not as though they had to go in order and that they only went one at a time. By the time we get to the third line of defense, the humoral and cell-mediated immunity with the T cells and the B cells, every aspect of the immune system is now working, all right? It's all now working together, wiping out bad guys like crazy, all right? So complement fixation, antigen binds to the IgM or IgG, antibody changes shape, initiates complement binding. Once again, we're going to see there are different kinds of antibodies. The last slide we talked about the IgDs. Now we have IgMs and IgGs. So they are going to do the complement fixation and get the complement proteins involved. This primary defense against foreign cells and bacteria, all right? Then agglutination. So antibodies have 2 to 10 binding sites. So look in the lower right illustration. Well, in the lower left, you can see an antibody that has 2 binding sites, 2 receptors. But look in the lower right. The IgM antibodies have 5 arms, and each arm has 2 receptors. That means they can grab up to 10 bad guys at a time. So here you see bacterium there, bacterium singular, bacteria plural. Here you see IgMs that are grabbing a hold of bacteria, and look what they're doing. They're cross-linking them. You got one bacterium that's been grabbed by both IgMs in that picture. And so what's going to happen? These IgMs are going to be grabbing bad guys like crazy. That's going to cause them to clump together. Agglutination. Same thing this is what happens when you have a transfusion reaction, all right? This is what's involved with the transfusion reactions. If you have, because the antibodies for the red blood cells are IgMs. So look at what's going to happen if you transfuse the wrong blood. These IgMs are going to be grabbing red blood cells like crazy, agglutinating them, and then causing giant blood clots. Well, that's very bad in transfusion, but if it's actual bad guys, this is great because now it's going to grab all these bad guys, these bacteria, and then that will allow other cells to come and kill them, all right? Remember, the antibodies are just handcuffing, but here they're going to handcuff big, giant clusters of them. So when, the eosinophils and the natural killer cells and so on show up, the neutrophils, they're going to have the advantage of having whole bunches of bad guys all in one place where they can just kill whole bunches of them all at once, okay? Binds to multiple enemy cells, clustering them and immobilizing them so that they can all be killed in one giant bunch, all right? And then precipitation, what's going to happen is they may actually precipitate out of the fluid. You're taking a chemistry class, you know how you can form precipitates? You can mix 2 liquids, it looks like. It's got chemical molecules inside. And when you mix them, suddenly it starts forming solids. Yeah, the same thing can happen here. Once you agglutinate all these bad guys, they may actually precipitate out of the solution. And then things like eosinophils, they're an easy target for eosinophils at that point. create antigen-antibody complexes that precipitates, and then they're phagocytized by eosinophils, all right? There you go, humoral immunity, just like I had those diagrams before. Take a few minutes, go through this. These are really good slides, all right? They're taking the stuff I've been talking about. This is just the way a particular book, a particular artist decided to portray it. You would learn a lot by spending a few moments going through that illustration, all right? There's another one, all right? Some of you are wondering how to improve your grades. This is one of the ways. Spending time on this. This is what studying is. Studying is actually sitting down and taking time and working through it. And if you don't understand it, then you keep going back and forth between slides. You keep trying to figure it out. And eventually, you know, the little light bulb goes on. And this is what studying does. Studying really makes it, it plants it firmly into your brain. It really sticks. The understanding really comes about, and then you really grab a hold of it, and then you really know it. And that's how you do so well on exams and stuff. So here you see a B cell, a normal B cell, and then you see a B cell after it's turned into a plasma cell. What's the big difference? Look at what do the plasma cell have bunches of that the regular B cell doesn't have bunches of? You see? What's there? Yeah, look, mitochondria and rough endoplasmic reticulum. This is why you had to take Bio 156 first, remember? What do mitochondria do? They're making ATP because the plasma cell is making antibodies. Well, making antibodies takes energy, so you need lots of ATP. Therefore, this cell has a lot of mitochondria. And then rough endoplasmic reticulum, the rough ER, remember what happens in the rough ER? That's where you make proteins. And what are antibodies? Antibodies are proteins. Isn't this cool? All this stuff comes together, doesn't it? You can see now, it's just so cool. It's so immediately obvious. It just fits together so well. The plasma cell is going to have a lot more mitochondria and a lot more rough ER because it's making antibodies. It needs the rough ER to make the proteins, and that takes energy, so it's got the mitochondria to give it the ATP it needs. All right, which of the statements below is false? Plasma cells are differentiated B cells that make antibodies. Cytotoxic T cells respond to MHC1 molecules. Cytotoxic T cells kill infected cells. Antibodies bind to MHC2 molecules. Antibodies mask the pathogenic region of an antigen. Which one of those is false? Yeah, D. That doesn't make any sense. Antibodies don't bind to MHC2. Antibodies bind to antigens. The MHC2 is where an APC puts the epitope of the antigen that it ate and displays it to a helper T cell.